And I'm going to talk to you guys about my thesis, which is investigating the effects of low pH on the metabolic physiology of red abalone grown in integrated multi-trophic aquaculture. So just for some background, ocean acidification refers to the gradual decline in ocean pH. This is known to be caused by excess carbon dioxide in the ocean. And this is projected to have widespread impacts on lots of things, including aquaculture facilities, which are going to be pumping in increasingly low pH water, and especially those that are growing organisms with calcium carbonate structures, such as mussels, oysters, some seaweeds, and red abalone. So red abalone populations crashed along the California coast in the 1970s. This was due to things like overfishing, habitat loss, being outcompeted by urchins. You can see in this graph that even as of recent, the populations are declining and showing little recovery. Past studies found that low pH on red abalone stunted their shell growth. There was a varying respiration response. Some found no response, some found higher respiration, and there is evidence that it may increase oxidative stress. So we used an integrated multi-trophic aquaculture system that could help alleviate these harmful effects. What this basically means is we just have red abalone and our red seaweed dulse grown together in one tank. The way this works is that the water is pumped in directly from the ocean. Photosynthesis occurs in the seaweed, which consumes carbon dioxide and raises the pH. That water gets circulated, and then hopefully the abalone grow faster and healthier in this buffered water. This is a mutualistic system, so in it the seaweed provides the abalone with increased pH and oxygen and decreased nitrogen, and the abalone provides the seaweed with decreased pH and their nitrogenous effluent. So my three questions are: how will pH change with seawater tumbled in ambient versus acidified water? How will differences in pH levels affect the respiration of red abalone grown with seaweed? And how will differences in pH levels affect the oxidative stress physiology of red abalone grown with seaweed? So to answer these questions, we designed this experiment with 3 different pH treatments. We had our 8.0 treatment, which is our ambient, our control. We had our 7.5, which is the 150-year ocean pH projection, and then the 7.2, which is that same projection plus an upwelling event, which lowers the pH even more. We had 3 header tanks to set our conditions. 2 of those had carbon dioxide bubbled into them to lower the pH, and those fed into our 100-gallon IMTA tanks. 15 of those had 250 red abalone and 2 kilograms of dulse, and 3 of them just had seaweed and no abalone. And we held these conditions for 6 months. So to answer my first question, we had apex pH probes in all of the tanks. You can see on the left side just a general overview of the average pHs per treatment. And then on the right side, when we separated it between day when photosynthesis was occurring and night when no photosynthesis was occurring, we found that the pH was raised significantly in all 3 treatments. So this just means that our system is working like we think it is, which was great to see. To answer my second question, I took the respiration of red abalone after 2 days of exposure to our treatments, and then again after 6 months of exposure to the treatments. After 2 days, I found that the 7.2 treatment was actually respiring less than the ambient treatment. The y-axis on this is negative, so in the ambient treatment, the oxygen was being depleted faster. And this is the opposite of what we expected to see, and it might suggest that there's some sort of suppression going on when exposed to really short-term low pH conditions. After 6 months of exposure, we found no differences between any of the treatments, so this might suggest there's some sort of acclimation going on after long-term exposure. To answer my third question, I took samples from each tank on 8 different time points. 6 of those were within the first 16 days, and then the 1-month mark, and then at the very end of the experiment at the 6-month mark. And I wanted to isolate an antioxidant called superoxide dismutase. And this is produced in response to oxidative stress. So the idea is that if they're more stressed, they would have more of this antioxidant, and we could measure that as a measure of stress. I found that there was no differences between any of the treatments on any of the days except for the very last sample. At 6 months, the 7.2 treatment had significantly more superoxide dismutase than the ambient treatment. So what is this telling us? This is telling us that our oxidative stress and our growth data, which I included a graph of there, but if you want to know more, Jeffrey Osgood is right there. He's going to give a poster tomorrow on it. You should go talk to him. But these are telling us that abalone in low pH water are experiencing prolonged stress. And this could lead to sublethal effects that are hard to measure. Like it could affect their reproduction, it could affect the resilience to other stressors, and it might just have other ecological and commercial impacts. But the IMTA system does increase the pH of seawater, so this could help the abalone grow faster, be less stressed, and these are really robust systems that will be able to be used far into the future as ocean acidification gets worse. Thank you.